So our next speaker, we, for some reason, stay at least partially in the state of Tennessee, is Jens Meiler from both Vanderbilt University and from University of Leipzig in Germany. And we're switching gears, so we're now going from T-cell receptors to immunoglobulins/antibodies. And so Jens will talk about how to do antibody design and antibody modeling with the Rosetta software. So thanks a lot, Jens, for joining us, at least virtually, and we're looking forward to your talk.
Yeah, thanks so much for the introduction. I hope people can hear me okay. Then let's get started. Unfortunately, I couldn't make it in person, so I'm still in Germany, but hope to join next time. I will present some of our research on modeling immunity with Rosetta, methods for antibody and antigen design. So we do develop a software that's called Rosetta after the Rosetta Stone. Originally, it was written to predict protein structure from sequence. However, this software has developed into a framework for tackling all sorts of molecular modeling challenges, in particular when it comes to protein engineering. And so you can use it for really a substantial number of different tasks, including drug discovery, loop modeling, or looking at membrane proteins specifically. But it is also a very important tool for modeling antibodies, antigens, and their interactions. This tool is not developed in my laboratory alone. There is a consortium of more than 50 laboratories worldwide that develop and maintain this particular software. It is free for academic use, and there are about 20,000 laboratories worldwide that actually are using the software, tutorials how to get started, etc. So let us know if you are interested in this. And obviously, it's a very vibrant developer community writing new algorithms every day. And these people are distributed around the world. So what I will do today is I will try to give you a glimpse into some of these computational algorithms that are developed specifically in my laboratory, and then also show you some of the possible applications in designing antibodies and antigens. And so this little video, which I hope is visible, shows sort of a Rosetta design simulation. So this is obviously not an antibody or an antigen. That's just a small protein for illustration. And what I did here is I made this little movie that sort of allows Rosetta to find an ideal amino acid sequence for the core of this protein. So you see the algorithm is testing all 20 genetically encoded amino acids in different conformations to find one sequence and one conformation that combined give a low free energy and are therefore predicted to be stable. And in gray, you see the crystallographic coordinates for this particular protein. That's actually a protein that was originally engineered with the software Rosetta. So you can use it to design new protein folds if you like. Now, we're going to apply this to antibodies and antigens, and this is knowledge much more familiar to you than to my usual audience, but we do know that there is a large diversity in the antibody sequence space through the recombination of these VD and J genes, which give rise to this beautiful area of an antibody with heavy chain and light chain complementary determining regions 1, 2, and 3 that really can vary in sequence and also in length of these loops substantially, creating in three-dimensional space very different surfaces that can then bind to antigens, for example, to surface proteins of viruses, which is what I gonna focus on today. And even more amazing, while the human immune system has built in a large repertoire of these germline antibodies, it has also built in a mechanism to improve the affinity of those antibodies. So to have the immune system as flexible as possible, and also because the number of possible threats out there is much larger than the number of antibodies we can sort of sustain at one given point in time. There's this wonderful directed evolution kind of experiment in the immune system that allows the immune system to identify antibodies that bind to a possible target protein, multiply them, and add mutations to optimize the affinity, and then create high affinity antibodies to the target and even memorize those for future use. Now, we're going to work with Rosetta in the structural space. So here we see a co-crystal structure of PG9, an HIV antibody, a partly neutralizing HIV antibody, bound to a mimic of GP120 of the surface protein. And green, you see these glycans that sort of protect the surface of the viral protein. And you see what's particularly special and nice about this antibody. It has a very long HCDR3 loop, which sort of winds in between these glycans and has this little beta hairpin in cyan, forming a hydrogen bond network, backbone hydrogen bond network with the viral protein, which is thermodynamically very stable. And so this is one of the pod antibodies known against HIV amongst many others, and we will meet that antibody in some of the stories that I'm going to talk about in just a second. But I promise that I will try to explain why this field is so exciting. And it's so exciting because every day, new technologies are added to the Rosetta software to model these antibodies, antigens, and their interactions. And so one particular improvement in the Rosetta software that we added quite recently is a potential that mimics hydrogen bonds, salt bridges, but also pi-pi and cation-pi interactions that are often made in aromatic side chains. And these interactions are particularly important since they are frequent in interfaces between antibodies and their target proteins. So by improving the Rosetta energy function to more accurately model these particular interactions, we can actually have higher success rate when we predict the structure of antibodies and when we design these interactions. In a nutshell, for the chemist amongst you, to achieve this improvement, we actually add the optimized quantum mechanical optimized location of the electrons, and then use that position of the atoms plus the position of the electrons to optimize geometry. And that gives us a higher accuracy and also more simplified potentials than if we formulate them from the atoms alone. And so here we see one application that's not yet designing antibodies. This is structure prediction. Some of you might remember this particular story from 2013 in Science and Nature, where mutations in avian flu hemagglutinin were introduced that created a mammalian transmissible avian flu variant. And at the time, we had, with our collaborators, Jim Crow, who is director of the Vanderbilt Vaccine Center, and all of the projects I show you are collaboration between our both laboratories. He had identified very exciting antibodies that actually H5.3 in this naming scheme that actually neutralize this particular variant. So these are human antibodies. And the question that was brought to us at the time is, what's the structure of the complex? And we only had this low resolution density map and some hydrogen deuterium exchange mass spec data to guide us. With that data, we were able to fit a model of the antibody to hemagglutinin and determine the orientation, how it binds, so heavy and light chain orientation, which was ambiguous with this energy function that sort of told us it must be orientation B, because that sort of chimed with all of the mutational studies that were done at the interface of this particular interaction. So, but let's switch to actually not just predicting those interfaces, but engineering them. And we will again start with a more basic science question. And we do know that the sequence of germline antibodies is altered during this affinity maturation. So you can go to the protein data bank and identify co-crystal structures of mature antibodies that stem from the same germline gene combination. So that started out with the same sequence, at least in the regions encoded by these genes. So we were wondering what mutations did this affinity maturation process add to improve the affinity, and we wanted to ask this question in a computational way. So what we did is we took these three co-crystal structures. So down here we have the antibodies. They are co-crystallized with their targets, three very different targets. And what we did now is we ran a so-called multi-state design protocol. So instead of adding just one of these complexes to Rosetta, we ran a simulation with all three antibodies present, with all of their antigens present at the same time. And so here's a superimposition of the heavy chain domain. We see they differ slightly because of the mutations introduced, but in our simulation, we asked Rosetta design us one antibody that binds all three of these targets. And even without knowledge that of the germline sequence, we didn't tell Rosetta how the original antibody, the germline antibody sequence was. The sequence that Rosetta returned to us was very close to this germline sequence. So here you see this design simulation. I hope this is possible over the internet. So Rosetta is optimizing all of the amino acids. And the sequence that it returns to us actually was very close to the germline sequence. Interestingly, to arrive at this germline sequence, we not only had mutations in the interface, but also outside of the interface. And this was a theme that did not only work for this one example. We have in this manuscript several examples. When you run multi-state design, you get similar germline-like antibodies. And so we have basically this correlation here that if we do a multi-state design, it reverts the sequence back to the germline sequence. So it's an in vivo reversion of the sequence towards germline that goes along with our request to engineer a polyspecific antibody that recognizes all targets. And if we do the reverse experiment, if we run a single state design to improve the affinity, we get not surprisingly, a sequence that's very similar to the sequence observed in the mature antibody. And the conclusion from this particular experiment is in part that these antibodies are, germline antibodies are in part optimized to bind to multiple target proteins. If I design an antibody that is polyspecific, it tends to have a germline sequence. So there is some flexibility built into the sequences of these germline antibodies that makes them flexible and allows them to recognize multiple targets. And part of the mutations introduced in this affinity maturation process are maybe not even at the interface optimizing the enthalpy of the interaction, but they are freezing the antibody in a conformation that's needed to bind this specific target of interest. So it reduces the entropic cost of binding. Now we can apply this, and let's come back to our friend PG9 here. And PG9 is partly neutralizing. So with our collaborators here, we got data on binding and neutralization with respect to a panel of HI viruses. And this is the wild-type antibody, the concentration needed to neutralize, and you see it hits maybe about 40% of this panel. However, the sequence coming out of a patient, we have no guarantee that this is the optimal sequence for polyspecificity, right? This is not necessarily the best possible sequence for this antibody to be as potent and as potent as possible. It's the sequence that happened to be found in this particular experiment. But what we can do is we can run Rosetta design on this region of the antibody, on the HCDR3 loop, that and ask it to find mutations that improve the affinity. And we did that. We tested 5 different candidates, antibodies that came out of the Rosetta design. One didn't express, so you see only 4 columns here. And the first couple of columns are maybe about as good as the starting material. The last one is not really good, but the third column, this particular one, as a single point mutation, is about 4-fold as potent and twice as broad. So it hits substantially more strains, and it is also a lower concentration. It's usually 4-fold lower concentration on average that's sufficient for neutralization. And so we went on to determine the crystal structure of this asparagine to tyrosine mutation in the apo state. In the apo state of the original antibodies experimental structure, the HCDR3 loop was not resolved, indicating that it might have been more flexible. Now, in our modified antibody, this conformation is sufficiently stabilized to actually be resolved experimentally in this crystal structure, and also superimpose very nicely with our model of this particular interaction. So one of the next questions that one can ask is, now, if this is such a hard neutralizing HIV antibody, and we might want to target this class of antibodies with a vaccine, with an immunogen eventually, do HIV-naive people have precursor antibodies that can be converted with a vaccine into neutralizing antibodies? And so for this purpose, in the Crow lab, there was a sequencing experiment done at the time. 64 HIV-naive blood donors from the Red Cross in Nashville. Their antibody repertoire was sequenced. We got about 25 million unique HCDR3 sequences. About 25,000 of those had the right length, belong to this class of very long, this is 31 or so residues, very long HCDR3 loops. And now what we can do again computationally is we can test each of those 25,000 sequences, and we can thread it through the co-crystal structure of the antibody with GP120, and we can ask, how stable are you in this conformation, and what is your predicted binding affinity to the target protein? And so we went ahead and did that. We had to develop another computational method to do the screen in a reasonably fast time, because that calculation is somewhat computationally expensive. And on this slide, you see each dot is one representative out of the 25,000. I only show the top 50. On the y-axis is computationally predicted thermodynamic stability. On the x-axis is the predicted affinity to the target protein. PG9 is somewhere down here. Here's PG16. So PG9 is predicted to bind with a very high affinity and is very stable in that conformation. However, these 50 antibodies we found in these HIV-naive humans, some of them approach the predicted affinity, but none of them wants to adopt this very unusual conformation that the CDR3 group has.
But you have 5 minutes left.
This would also not be expected, since all of these humans are HIV naive. Now the question obviously becomes, can we, with Rosetta design, how many mutations do we need to add to achieve neutralization? So we again ran our design experiment, and now we see that the sequences of those antibodies move down into the region of PG9, and some of them start to bind to the target antigen, albeit with a quite low affinity. Now, 5 minutes out of only 5 minutes left, so let me jump a little bit ahead and tell you a few other stories. We can turn these long CDR3s also in small peptides and stabilize them and get high affinity binding. I will skip this particular story. This is all published. This is screening for Ebola and Marburg antibodies. I will skip that to talk just about one last project on epitope-focused vaccine design. Some of you will remember this particular paper, not out of my laboratory, but Jim Crow and the chief at the time, where they engineered with Rosetta a protein that stabilizes in particular peptide here, in particular epitope, in a way to allow it later to be used as an immunogen and elicit antibodies. And this was for RSV. So we repeated that experiment for the Ebola virus. Jim Crow found a particularly exciting antibody, BDBV223, that binds to the membrane proximal region. And we cut this antibody out and used Rosetta to engineer a small protein that sort of mimics this particular membrane proximal region, this particular helix, in order to elicit these type of antibodies. And our little immunogens here bind also to these antibodies now very tightly. We determined the three-dimensional structure to confirm that they adopt the right conformation and expose the epitope. And then we put them on a Rosetta engineered self-assembling platform so that we can display 60 copies of our immunogen and use this as an immunogen to test if we can elicit the type of antibodies. And story short, this actually works. We get a very robust expression of these type of antibodies with this kind of immunogen. And with that, I would like to acknowledge the laboratories in Leipzig on the top and Nashville on the bottom, and this particular in particular the collaboration with Jim Crow at Vanderbilt University and funding from NIAID. Thanks for your attention.
Yeah, thanks for the great talk. And we have time for a couple of questions.
Yes, thanks for your talk. For your amino acid optimizations, are you using rotamer libraries, or is there true backbone flexibility in your system? And if yes, are you using restraints?
It's a combination. Rosetta uses for side chains rotamers. For the backbone, it uses a pre-computed set of backbone fragments. These particular combinations focus on the side chains using rotamers and then also doing a small movement refinement. However, the backbone is not entirely restrained. It can move, but it can only move in very small in very small steps. So maybe 0.2, 0.3 angstrom. But that is important to accommodate some of those mutations. There are no physical restraints needed, just the structure holds so tightly together that the backbone wants to stay where it is.
Thanks.
There's one question in the audience. This study proves how few amino acid changes can be very important for binding affinity and antibodies. Can a similar study be conducted for T cells and help the field of TCRs clustering to understand the limits of similarity between the TCRs and their specificity from a structural point of view? Yes, I believe so. In fact, there are some papers out there. I'm not yet a real expert. We are just starting to do some of these calculations. But I definitely believe that this is a possibility.
Okay, if there are no further questions, then thanks again, Jens.